It's a real pleasure to have Dr. Nicolas Karakatsanis with us. He's a great former member of our lab, colleague and friend. He's a pioneer in quantitative multiparametric head imaging. He's a faculty member at Weill Cornell Medical College in New York City, and it's just a tremendous pleasure to have known him and to have worked with him. And thank you for accepting our invitation.
Thank you, Dr. Amin, for the kind introduction and for the honoring invitation to present this highly timely topic to your lab and the institution there. So I'm looking forward to our session. I'm going to be talking today about the value of explainability for the clinical adoption of artificial intelligence in nuclear medical imaging. There are no disclosures to state for this topic. The structure of my lecture is going to be mostly break down into major topics. The first one will be the classification or taxonomy of different explainable AI methods for biomedical imaging, including basic and clinical research in radiology and especially nuclear medicine. And also, what are the challenges and opportunities of explainable AI applications for that context, to facilitate the wide clinical adoption and impactful outcome of AI in global healthcare, in particular nuclear medicine. So explainable AI is all about increasing the transparency and trustworthiness of AI models. This is particularly important for the healthcare domain and the nuclear medicine domain in particular. So we're going to discuss what is the added value of adding explainability features in AI models, and how this can enable translation of the AI benefits in radiology and global healthcare. As an introduction, as we all know, deep learning in the recent decades has demonstrated remarkable performance for both diagnostic and theranostic imaging for various diseases and modalities, and has demonstrated high potential to be used in the clinic extensively. AI models are usually evaluated in terms of their predictive performance, for example, in classification accuracy. However, the performance metrics do not necessarily capture whether the evaluated model is right for the right reasons. Sometimes something can be right, but for the wrong reasons. So in explainability, we try to evaluate how valid the reasons are that led us to a correct decision. Machine learning models can replicate biases and other confounding patterns for the input data, but these don't necessarily have to be discriminative or not. So, for instance, in COVID-19 detection, we have seen that it can rely on markers, edges, other features such as arrows that are clinically irrelevant annotations. So, but they can still result in correct results. So this is the so-called clever hands behavior that may result in a classifier's right decision, but for the wrong reasons. And another term known for this effect is the shortcut learning term. In current practice, we have seen low deployment of such algorithms because of their lack of transparency and black box mechanism, which is really something that discourages clinicians for their extensive use nowadays. Therefore, explainability can really help to close the gap between the medical professions and those algorithms. So in this lecture, I'm going to make some introduction about the taxonomy and basic features of some of the most established explainable AI models in the context of biomedical imaging. I will present some examples from MRI, CT, or PET, and I'm going to discuss challenges, opportunities, and future suggestions that can maximize the clinical benefit of their application in radiology. So computer-aided diagnostics have recently, the last few decades, started extensively using deep learning methods. The advantages are obvious. Can have similar or superior performance in comparison to medical professional aided diagnostics, and therefore they are characterized by great potential for the clinical workflow improvements. However, despite those promising preliminary results, deep learning has not been widely deployed clinically, and despite the fact that it can extract features in an unsupervised manner without already defined assumptions and regulations, and without requiring manual extraction of features, depending on VOI annotations and so on, which can be cumbersome. The reason for that is the lack of trustworthiness, and explainability is the ability to understand deep learning model decisions and their reasoning. For that, it requires efficient learning and explainability to work in synergy. It's not enough to train a network and learn, but also to understand how it learns and why it leads to those decisions. So the lack of transparency and opaqueness is what has actually limited its adoption and what explainability tries to address. Deep learning transparency should be improved, as I say, as we explained before, in order to close this gap. And this will provide medical professionals and stakeholders with a more pragmatic explanation of the model's decisions and the process towards that decision making. They can mitigate AI opacity by providing the attribution maps of the explainability or attribution of the methodology by which the result is provided. And this is what we call what is so-called attribution maps or feature important maps or heat maps. And I'm going to be referring to those terms more extensively throughout the talk. But basically, explainable AI methods try to attribute the models to some plausible explanations. And the most standard way to do this is by backpropagating the model decision to target specific attributions present in the image. Having said that, we realize that explainability AI may add another target for our AI models, not just to complete the task, the segmentation, the classifications, and so on, but also to provide new imaging biomarkers in the clinic, because it can function as an indicator of normal or pathogenic biological processes by indicating the reason behind those decisions. So it can actually complement medical professionals in their decision-making, and it can monitor and assess responses to an exposure or therapeutic intervention in addition to the task involved underlying. So explainable AI methods should provide transparency about quality and legibility of a decision, explanation, and associated errors. Explainable AI must therefore cross translational gaps and overcome those gaps, both in technical and clinical domain. And also this will assist with improvement in the cost effectiveness of the deployment of those methods. For instance, the new European Medical Device Regulation has endorsed strict transparency regulations, which call the need for more effective use of explainability in clinically approved, regulated AI models in healthcare. So I believe that explainable AI is something crucial, is a new component, a very crucial component for the more transparent, ethical, safe, and trustworthy deployment of deep learning algorithms in clinical practice. And it's something that we should start considering as an important feature in every AI model that we try to build if possible. However, it's important to understand that this has to be done in parallel to better understanding of the current practice of each task that we're trying to target with AI technology. So here's an example of the different steps throughout radiology clinical workflow. We start with an investigation of organs and imaging devices that allow you to inspect those organs. This will result to images and to data analysis of results. And at the end of the day, the doctor will collaborate with the data analysis software and the data to provide diagnostic, prognostic assessments. But another important element in order to increase the trustworthiness between the data analysis, the AI, and the doctor is the explanation ability for the reasoning behind those data analysis. So explainability can be valuable and crucial for all aspects of radiology healthcare, both for physicians, scientists, and technologists, for the actual patients themselves, for the healthcare administration, but also to help further improve and debug AI models and increase their generalizability. So it can also benefit the developers of AI models as well. So in terms of the classification of the models, For explainability, I would say there are 4 different major classes that we could use. We can classify explainability methods based on whether they can be applicable for a specific model or multiple AI models and architectures. So you can have specific model or agnostic model explainability methods. You can also classify them based on whether they explain local voxels only or also can explain interdependencies across voxels and organs. So you can have either local or a global model AI explainability AI methods. You can also classify them based on their ability to explain the model intrinsically as it is learning, which is an ad hoc method, or as a surrogate model that is provided afterwards to explain the model itself. So in that case, we are referring to post-hoc XAI methods. And this is actually most of the methods that have been established so far. Recently, we also had ad hoc methods, which are even more effective and promising. But the most standard approaches we have so far in clinical practice and in extensive validation is for post-hoc methods. And the other classification is whether they can actually provide attributions for voxels only or for a group of voxels. If it's on a voxel level, then it's high resolution. If it's on a region of voxels, then they're not very specific in terms of spatial domains, so they're characterized by low spatial resolution. Here's what I already explained. So let's start with the post-hoc methods first. One example for post-hoc methods that first was implemented for explainability is for classification AI models. These are post-hoc methods that do not intervene to the learning ability of the AI model itself, but they are added as a surrogate model to provide an explanation for the original AI model decision reasoning. The majority of deep learning algorithms supporting explainability in biomedical imaging actually employ post-hoc methods for that reason, due to its wide availability, plug-and-play development. Post-hoc methods can be divided into gradient propagation methods, perturbation methods, or more traditional segmentation radiomics methods that will be briefly discussed later on. Here I have a table that has been provided by a nice review paper by Debrier and others in 2023, where they actually made a nice review of different explainability methods for radiology and nuclear medicine. So I highly recommend going in that paper, and I'm going to also refer in several slides to some of the findings from that review paper. So you can see here an example of post-hoc methods and how they're scored in terms of their different aspects of performance. So the first column is target specificity. The second column is the performance as a function of the resolution, whether they are high or low resolution. The green color reflects a high performance, the orange and medium performance, and the red color reflects low performance. Then the third and the fourth column is about local and global voxel dependency. And the last two columns refer to the model specific, if it's a model agnostic or model specific model, and about their technical complexity and computational burden when they're implemented. So these are 4 criteria for performance, and they nicely provided a rough estimate of their performance for all those different aspects. So you'll see here VG means vanilla gradient, deconvnet is deconvolution network, and so on. So we're going to talk a little bit about each one of them in the next few slides. So let's start with the vanilla gradient post-hoc explainability method, which is designed to create an attribution map by calculating gradients over the layers using a single forward and backward propagation. So basically, you have an input image, you feed it into your AI model to calculate an output score, as you would do always. And so a forward propagation method, but subsequently you have an additional process where you go backwards and you estimate the dependence, the gradient between the neuron convolution layers and the output, the calculated output. And so that's through a backward propagation method. And this will at the end result in the creation of an attribution or features map. which will explain you how each voxel is contributing to the decision and the reasoning behind the decision of each AI model. So this is the most basic approach. Here is a mathematical description of the process, where basically we want to optimize the L2 regularized image that maximizes this probability of predicting class C based on an input image X using a regularization term lambda. So here's another workflow of how this can be implemented for a few examples, such as for a brain imaging task here. You have, for instance, the convolution neural network with the convolution feature maps, and through the process of average pooling and then the fully connected layers, you end up with a classification decision or a segmentation result. And then you can backpropagate through the gradients of each layer to output a feature map here that gives you the probability of resulting to a particular classification result for every voxel. So you can use that as a heat map and as a salient map to explain visually how the network resulted in this decision about classification, segmentation, and so on. So the vanilla gradient is, as I said, one of the most established and intuitive attribution methods with low computational power. However, the cons is that it generates noisy attribution maps, suffers from gradient saturation, which means a change in a neuron doesn't necessarily affect the output of the network and therefore cannot be quantified. It also lacks the ability to differentiate between classes, so it's not class discriminative, as we'll see with other methods. For example, it cannot discriminate between healthy versus disease. It just gives you the probability that this will be the outcome for a particular task, such as classification, but it doesn't really discriminate between the different classes of that process. A Neuro XAI framework that compared different explainability methods for neuroimaging has actually found out that vanilla gradient exhibited noisy attribution maps and gradient saturation, both for classification and segmentation feature visualization. That was done for MRI analysis of brain tumors. In another example, from CT images to enhance the contrast, we've seen similar results for vanilla gradient performance. The attribution maps, again, were noisy, and they were suffering from the effect of gradient saturation. So another approach is the deconvolution network approach, which is effectively equivalent to vanilla gradient, apart from the way the gradients are calculated. We use now rectified linear unit function, which will output only the positive input values that are used for the feature calculations and the attribution map generation. Excuse me. So this is basically going to be using, again, a reversing pooling component, reversing filter component. So it will reverse the forward propagation process. But with the convolution approach, we're not actually training that network, but we use it to probe our original convolutional neural network with it. So we can attach these layers in our original CNN layers. And here's an example where on the right, you see the layers for a CNN network. And on the left, you see how the deconvolution network layers are attached to the respective CNN layers. And the reverse process is followed in order to backpropagate the gradients and through the deconvolution process, estimate the attribution maps. So this is another example here where you can see the max location switches, which is when the two networks are attached to each other, and the process is reversed from pooling back to unpooling until we get the original features that are most important for the decision that our network output. Again, for the deconvolution network approach, this helps with improving the model convergence with respect to the simple vanilla gradient method, because it uses the rectified linear unit function that only outputs positive values. And the results are less noisy and more human interpretable because we can focus on more specific regions. However, we still have absence of pooling layers, which can result in non-image specific explanations, and therefore the generated heat maps are less informative. And this is also part of the reason for that is also because we are rejecting negative pieces of evidence during the back propagation. So there was a framework that actually was published comparing different DL algorithms and their attribution methods. And one of the methods that they used for attribution for explainability was the vanilla gradient and the deconvnet method. And they found that they both had some decent human interpretable results. However, they were relatively noisy performance with respect to other models that are used that I'm going to discuss later. So another approach is the guided backpropagation, which basically incorporates both vanilla gradient and deconvolution network in one framework. So as we know, as I tried to explain before, vanilla gradient accuracy is limited because of the flow of negative gradients. This is addressed with the ReLU function in the deconvolution network approach. But the guided backpropagation approach actually combines vanilla gradient and deconvolution network. Basically, it guides the vanilla gradient backpropagation process with the help of the deconvolution provided by the deconvnet approach. So basically, the deconvnet approach, as we said, uses the rectification component, which masks all non-positive values, and then ignores the input and uses the top signal as a base in order to calculate the features. On the other hand, vanilla gradient uses all positive and negative values and focuses instead on the gradient values computed that based on the input image. So the guided backpropagation is actually combining those two by masking the rectification layer and applying it on the vanilla gradient values. This will result in the removal of some degree of noise from the negative gradient values, and therefore noise removal will also be observed in the final feature maps that are generated with the guided backpropagation method. Here's an example of an original image and the features that result in the proper classification of that image. You can see on the second column the deconvolution results by themselves. On the last column is the vanilla gradient results, and on the third column is the combination of the two, where you can see that you have less noise and more specific image-specific feature maps that help you better understand the features that really contributed to the decision about the classification of this image. So you can see how you can combine both best of both worlds with guided backpropagation to come to a better outcome in terms of explainability and interpretability of that explainability features. Again, this doesn't come without a negative, without cons. So the maps that are generated, as we saw here, they're relatively sparse and therefore not useful for complete image characterization. However, they're less noisy compared to VG or deconvnet alone. And there were also studies where they actually describe those methods, and they have shown clear advantages of using these methods for their applications. I'm gonna leave those details to you to read it and later if you want, for the sake of time. Another method that has been used is the layer-wise relevance propagation method, which actually uses relevance scores and propagates them backwards over the neural networks using specific layer relevance propagation rules. And more particularly, I'm referring to the conservation of interneuron dependency. That means that what has been received by a neural layer will be redistributed to the following lower layer in equal quantity. So they actually may make sure that this concept is satisfied across the backpropagation process. And here is an illustration of that concept where you can see how the content of a neural layer is redistributed backwards to the lower levels, lower layers. And here's an example of relevance maps, so feature relevance maps that have been used for predicting abdominal aortic aneurysm. And you can see when they're superimposed on the CT images for two example cases, how well they match the aorta. And provide better specificity in terms of spatial domain. Again, this method has the advantages of propagating relevant scores between the neurons, therefore addressing potential gradient saturation issues that we have with VG or deconvnet methods. However, it also has demonstrated low specificity in attributions because of the low, when the deep learning model itself is underperforming, this will affect the performance also of the explainability method. Here's examples I'm referring to from the abdominal aortic aneurysm study that I showed you before. And they are showing that the performance of the method can be sensitive if the DL model is biased. That will also affect the results of the attribution method and the accuracy. So we have to be careful also about the quality of the deep learning methods that we're using to evaluate explainability. And these are other examples from the COVID-19 classification study and also from other studies where they have shown that we do improve class discriminative explainability and precise localization of lesions using this method. Another method that has been used is the deep learning important features or deep lift method. This one employs a neutral reference activation. So to describe the change of a new neural activation relative to that reference. So for example, if we know the neural activation for a normal healthy CT scan, then we can see what is the changes when we use a pathologic CT scan for that activation. And this can be incorporated when we calculate the scores and the attribution maps to better assess the clinical relevance of our results, to improve the clinical relevance of our results. So this addresses the saturation problem of other XAI propagation approaches. However, this depends on the internal wiring of the network. This is the same with the LRP method also. It's also depending on the internal wiring structure of our network. So we can have two identical models with different internal wiring, which could produce different attributions. So even though they're identical, they will produce different attribution results, and that's not desirable. So it is not implementation invariant. Here's examples where they have applied the deep lift and LRP for identifying multiple sclerosis in patients using MRI. And they have shown that deep lift performs slightly better than LRP, but they both perform much better than vanilla gradient methods. One more classic example of explainability method that you might have heard of is the class activation mapping method. That's one of the most well-known model-specific attribution methods. I call it model-specific because it's specifically designed for CNN convolutional neural networks. So it's not model agnostic as some of the previous methods that we discussed. It employs a global average pooling layer instead of multiple dense layers. And the gap layer introduces linearity between the last convolution layer of your convolutional neural network and the final dense layer. So the drawback, of course, is that it uses the last convolution layer, which results in low dimensionality for the attribution maps. But the good thing is with CAM methods, you don't have to go backwards. So it's less computationally expensive from that aspect, provided, of course, your features are not many, because if you have many features, then you have to retrain for every feature, and this will increase computational burden. So this can visualize whether a model is able to roughly focus on specific targets. So the main advantage with the class activation mapping is that it's class discriminative. So you can actually see how for every class, how the attribution maps are formed, which is very important. But on the other hand, it's also suffering from low specificity, and the general method, the simple CAM method, also the discriminative power is not very high as it is with the GradCAM methods that I'm just going to show you later on. So they may exhibit inferior performance versus more general networks, which we're going to talk later on. And here is an example of a perturbation analysis where they did multiple explainable XAI methods evaluation, and they showed that gradient methods have higher specificity than CAM, but CAM can be more discriminative. The more generalized version of the CAM approach is the gradient-weighted CAM method. And here you can see an example of that method as it was first presented. And you can see that now we're using the gradient of the output of the network with respect to the activation of the feature maps. So you actually weight your feature maps based on those gradients, and then you take the global average of those, as you can see in the graph here, and this will result in a more accurate class activation map and more clinically relevant. However, as you see, it still lacks from low resolution. And it's quite blurry heat maps, as you can see on the right. So it can improve the accuracy and interpretability of results. It's more class discriminative. Unlike CAM, there is no requirement for specific CNN architecture. It still requires some type of convolutional neural network, but it's not specific to a particular architecture. And the limitations include the lack of robustness when we're changing input images, and there's unclear explanation of the basis for prediction in complex images. So if you provide complex images, it will not perform as good as with simple images. And also it suffers from low specificity and resolution due to the low dimensionality of its attribution maps. So there are active development and research on GradCam methods, and I'm going to show you also a few examples in the few next slides to improve those limitations. So the gradient weighted CAM, as an example here, we have a whole image segmented using lung CT-based COVID detection, and we've seen that the method performs produces very specific attributions, but also non-specific attributions in some other areas. So it really helps when we are a priori segmenting the whole CT image to only the lung, then the performance of the method is improved significantly. So the take-home message that I want to highlight here is that it's very important that we can, if possible, we can reduce the trainable features or non-informative features of the image by performing, for example, some a priori segmentation. For instance, in the case of the lung, we don't have to provide the whole chest CT, but the segmented lung regions. This will really help not just the deep learning method, but also the explainability methods that are used as a post-hoc step to explain the deep learning method. So here's examples where they highlight this advantage and how by segmenting and preparing the data, it really helped the performance both for the deep learning method and for its explainability afterwards. Another example of a generalization of the GradCam method is the GradCam++ method, a feature importance weighted GradCam method, which takes actually the weighted average of the gradients that I was referring to before. So the weighted is determined by the importance of each feature. So you take also into account the importance of the feature. The caveat here is that you don't only calculate first order gradients, but now you require higher order gradients. And this is a little bit more computationally intensive, but it has shown to improve better localization for the target specific features of the original GradCam method. And there are examples like in knee osteoarthritis using MRI where they've demonstrated this improved performance. And there is also the combination of the previous guided backpropagation method that I had described you with the GradCam method. If we combine those two by point-wise multiplication of their feature maps, we can obtain feature maps that are both highly class discriminative and high resolution. So we can get the best of the two worlds. And here is an example I'm going to show you here where they actually I first presented that method, and they've seen that by combining the guided backpropagation output, which is not very image specific, it's more general, with a more specific GradCam but low resolution image, we can obtain a guided GradCam, a feature map, which is specific both and high resolution at the same time. So it's one of the most popular combinations that I've seen for explainability methods in medical imaging by combining the GradCam method with the guided backpropagation method, with a straightforward combination, if you ask me. Another important thing here is to understand that a low performance doesn't necessarily mean that your explainability method is underperforming. This can be due to the DL algorithm, deep learning algorithm, being not performing very well. It will also be because of lack of anatomical specificity in the input data. For example, an unsegmented chest CT, if you segment the lungs, it may be considerably better performance. and also input image artifacts. So I would like to highlight the importance of using high-quality input image data, apply data minimization whenever possible, and employ an a priori quality control method to detect bias in the input images before processing. Another example of a post-hoc method is the occlusion mapping method, which also uses feature maps, but it uses a random perturbation method conditioning over the image. So it's more straightforward and more close to what a human would have done to explain a prediction method. So you basically occlude different voxel patches regions by sending input pixels to 0, and then you're trying to see if some voxels over the others or patches or regions can better explain the decision and attribute to the decision of the model. So in contrast to previous methods, occlusion maps do not take the feature maps into account, but only the different patches. However, those patches can be defined in a grid or atlas-wise combination of multiple voxels that are clinically relevant. So this doesn't necessarily is a negative aspect. So there are different variants of how we hide and reveal different feature importance part of the image. This can be done through a randomized input sampling approach. This is what the RISE method is using, where we generate multiple random perturbation maps, and we use point-wise multiplication of those. Or we can use square grid divisions of the input image. So this is a simple and intuitive method to perform and interpret, can easily be adapted to specific occlusion analysis and compared with traditional clinical anatomical atlas-based analysis. This will help transparency and intuitive visualization of the deep learning algorithm. However, it's too rigid to follow anatomical pathological structures present in the image, and it can require large computational requirements due to the many forward and backward propagations that are maybe needed. And they've applied this for predicting and visualizing Alzheimer's disease, and they've used, instead of rectangular patches, the Harvard-Oxford cortical and subcortical structure atlas, which really helped the performance of the occlusion mapping explainability method. Another approach is the local interpretable model agnostic explanations. This is by perturbating superpixels instead of patches. So superpixel is a group that follows a group of pixels that follows an anatomical pathological structure in the image. So it requires some clinical relevance, but it can really help with better explaining by incorporating that clinical knowledge in the process. And here's examples of those super pixels and how they can be used to explain classification performance. Lime has also pros and cons. It can provide a better representation of the image than the predefined occlusion mapping. However, by using superpixels as a whole, the occlusion maps have relatively lower specificity because they're bigger and have less non-target specific activation. And also there are certain parameters that need to be optimized during initialization, which can be difficult. And they've also evaluated this method for CT-based COVID-19 detection. They selected superpixels that followed the anatomical structure of the chest CT image, resulted in non-target specific CT features, and showing high activation, suggesting low specificity and non-target specific activation. So the last method that I wanted to talk about, the post-hoc method, is the SHAP method. This is one of the most advanced explainability AI methods. SHAP values are mathematical formulas based on game theory that are used to estimate the marginal contributions of all factors that could affect a prize in a competition. So they really represent each voxel's attribution to the change of the expected model prediction when conditioning on that voxel, and they use reference samples for that purpose. Deep SHAP is an extension of the SHAP method. Basically works in similar way like the deep lift method that I described to you before. It's incorporated into the deep learning network, and can provide both local and global explanation based on those individual pixel voxels input. And it can also inform whether a pixel or voxel is negatively associated or positively associated with a predicted class. The negative association is very important sometimes because it can help you discriminate counterfactual from positive factual points in the image, like points of the image where you can be adversarially affected by decision versus points in the image where they are actually contributed positively to the decision of the image. So again, they are powerful in providing both local and global interpretability. However, they may be difficult to interpret due to the presence of both positive and negative associations. They require reference samples, which are not always available, especially for non-rigid anatomical regions in the image. They're computationally expensive, and they require additional analysis and complicated analysis. So, moving forward, this was so far we talked about this category of methods, but there are also probability maps that can be used as attribution maps. This is when we have voxel level annotation, not just one label for the whole image like the previous approaches. In that case, for instance, for segmentation deep learning algorithms, we could use probability maps to come to a decision, like to segment a region in an image or a lesion. And they have actually shown that inherently the segmentation algorithms can be also explainable, because you can actually see the result of your segmentation and see how close it is to your desired region. So in this case, the segmentation algorithms are self-explainable and they don't need an additional post-hoc method to be explained. Radiomics is another example of features that can be used to explain the decision of an algorithm, because they've also been associated with a certain clinically relevant information. So another example is deep feature maps, which are attribution maps that are generated on the intermediate layers of the DL models. Of course, this will directly visualize the underlying feature extraction mechanism, but they can be sometimes hard to interpret. And we also have the physics-based AI models and clinical data approaches, or for instance, by using clinical data to inform our network and physics. This by itself increases the trustworthiness, because we know we rely on some physical properties that we have validated before on some actual clinical data, like the patient history. This could also may aid in better performance and transparency of AI algorithms. So these are self-explanatory methods. All those previous post-hoc methods have also some limitations. First, they require an extra model and extra training. They require reverse engineering of an already trained predictive model. So they require additional work and additional resources to generate the heat maps. And those heat maps do not necessarily explain the full reasoning process. They only give an intuition, making them sometimes irrelevant to the tasks in a more realistic scenarios. So they have not always fulfilled clinical requirements to correctly explain a model's decision process. A more ideal scenario would have been to use ad-hoc XAI methods, where the models are intrinsically able to both learn and explain at the same time. In this case, the deep learning models do not predominantly focus only on learning, but also require a method, a process that through learning, they can also explain what they're learning, to explain their behavior. So you can see these two approaches here, the post-hoc method that relies on backward and forward propagation with the original deep learning method, and the ad hoc method that directly outputs the attribution map from the deep learning method itself inherently, without having to rely on an additional method. There are examples of such methods, such as the XDNN and PIPNet that I'm going to talk to you, or the capsule networks, which involve subcollection of neurons. But I'm going to briefly refer to those. These are very new, very recent work on that, and there are not many applications so far in medical imaging. But I'm going to refer a little bit on those. So the first one I would like to highlight is the explainable deep neural network approach. And this employs a prototype identification layer in the network. So if you have already some predefined data samples, you can compare the new data samples based on their similarity to those prototypes. This is very human intuitive, and actually has shown some promising results so far in other application domains in imaging. So I think it will be very interesting to see how it applies also to medical imaging. So it's more transparent, intuitive model explanations, because we humans also have learned to extract features based on previous experience. So if we can provide those prototypes accurately, then this method can be promising. However, Again, selecting those representative prototypes per class can be a difficult task, and especially if we have a wide variety of disease morphologies. And also, difference in class morphology is not always trivial. But at the same time, those tasks, if we are successful on those class representations and prototypes, then this can be very powerful and class discriminating. There have been some examples for COVID-19 screening artifact detection, where they've used representative prototypes to assess new images based on their similarity. Another approach similar to the previous is the patch-based intuitive prototypes network. Is the next generation inherently interpretable models. They adapt the concept of recognition by components. So they analyze whether patches in an input image are similar to learned human understandable prototypical image parts. So they permit understanding of its reasoning based on those prototypical parts. And if they're out of if the prototypical parts cannot explain it, then they abstain from the decision. So they have certain potential because their decision-making process is in line with medical classification standards. They use prototype model that does not require any annotations or image level class labels. It's unsupervised pre-training of prototypes. So you don't have to deal with data quality problems such as undesired text in an X-ray and so on. And humans can manually correct afterwards the reasoning of the network of the explainability network by directly disabling undesired prototypes. So it's quite human intuitive, but really relies on having the right type of prototype components for this. And here are examples where they've used this technique for correcting medical image classification with PIPNet. You can see for hand-specific hardware, for shoulder prostheses, for joint disease in finger, and for various metalware, how these prototypes, which are relevant to the abnormal class can be visualized with a set of image passes. And I think this would be one of the future methods that will be widely established and advanced in the field for explainability purposes. The current challenge is to improve the alignment of the PIPNet with the domain knowledge, of course, and choosing the right prototypes. Another example you may have heard of is the so-called capsule networks. These are also networks that use vectorized representations. So instead of having nodes and blocks, here we have capsules. This is the new building block of those networks. And basically, these are neurons that are sub-collections of neurons at a layer describing the presence and the instantiation parameters of them, like orientation, thickness, skewness, and position of a particular object. For instance, a lesion, such as a tumor, or an organ, such as the lung. And therefore, they can vectorize that description and that representation and define it as a capsule of a sub-collection of neurons. You can see examples of that on the top right, how they define this orientation and positions for the different capsules. So you can also, so CapsNets basically is the network of capsules, and they encapsulate different pose information, different poses, for instance, with other instantiation parameters such as orientation, thickness, and so on. And so they're not just deep in height, they're mostly deep in width in this sense. So capsules from a lower layer try to predict the output for the higher layer based on those instantiation parameters. And lower layer vectors with high agreement are routed to the following layer. However, the other vectors that don't have a high agreement are suppressed. And this can result in a target-specific attribution map creation, which is self-explainable afterwards. Mixed caps is a combination of cap networks. So it's an extension of traditional capsule networks. And instead of a single CNN, we can have a mixture of different higher expertise CNNs together, each of which is specialized on a subset of the data, and an ensemble of a capsule network is created in the end. So again, capsule networks have their pros and cons. The pros is that this is a new trend in deep learning that can eliminate the pose and deformation challenges that we're facing currently with convolutional neural networks. They require less data and less computational power. They have better handling of different visual stimulus and better understanding of proportional changes compared to traditional pooling methods, such as the max average pooling methods that we use in typical traditional CNN networks. However, it's still a very early technology. It has not been fully explored, so we don't know its full potential yet, and further understanding is required before we deploy this to a more extensive sense. Moving forward, another example is attention mapping. I guess you've heard recently about that more and more. This is also an ad hoc method that can be self-explainable, employs an end-to-end trainable spatial self-attention mechanism where we train during, so basically here we have training of the feature extraction process during the actual model training. So we can do both learning and explaining at the same time. So this replaces traditional non-learnable pooling operations again. And the main novelty here is that they use attention estimators to compute attention mask from convolution layers as a goal to extract those feature vectors. They can classify input images using only weighted combination of local features, weights that are represented by these attention maps. So attention mapping is also investigated combined with multi-instance learning. This is another concept that tackles the downsides of weak labeling and labor-intensive per voxel annotation that we have in traditional CNN methods. We're not going to talk too much on this because we're running out of time. I'm just going to talk also about the need to determine the method that we're going to use based on the disease that we're targeting. All those methods that we just described before, like the post-hoc, ad-hoc methods, the model agnostic or model-specific methods, they have to be chosen based on the disease characteristics. The spread of the disease, is it localized or diffuse? If it's localized, we can use a local method that doesn't need to characterize voxel interdependencies. But if it's a disease that is spread throughout the body, we need to choose a method, explainability method, that can explain intervoxel dependencies across voxels, across organs. So these criteria have to be taken into account. Again, explain AI rising in popularity, and therefore it's important to evaluate the explanations themselves. So they have developed criteria to evaluate how well the explanations that are provided are high quality or not, are non-binary or not. So there has been this effort, this initiative, the Co-12 initiative, where they have defined 12 conceptual properties serving as a categorization scheme for systematically reviewing the evaluation practices for explainability AI. And here's an example of what characterizes a good explanation. How this description of the reasoning is correct, complete, consistent, continuous, contrastive, covariate, and so on. So you can have all those criteria and use those in order to evaluate an actual explainability method, the quality of the explanation. This becomes more and more important as this is applied to the healthcare and medical imaging field. Again, as a summary, I want to say that one important challenge with deep learning is the lack of explainability, lack of interpretability of DL algorithms. This leads to the black box nature of this and the opaqueness, which is undesirable, especially in the healthcare domain. This leads to lack of trust, and lack of explainability is a major concern in the healthcare domain. So explainable AI can help you address that gap by providing interpretable models without sacrificing accuracy. The aim is to explain how a DL-based method can arrive at this decision by providing saliency attribution maps and highlighting the most relevant features that contributed to their decision. An important note here is that explanations should align with the medical knowledge and supported by clinical evidence. We believe that by improving the interpretability of these models, clinicians can better understand and trust the decision-making process, improve patient safety, and lead to more efficient and effective diagnosis and treatment of the diseases. So the acceptance, proliferation, and widespread adoption of a system are directly proportional to the trust in that system. Therefore, this is the objective of artificial intelligence, of explainability AI in artificial intelligence, is especially in the domain of biomedical imaging and radiology, is to increase trustworthiness, reliability, encourage collaboration between humans and AI methods, reduce bias and discrimination, ensure compliance with regulatory requirements. So while exploring AI, explainable AI can be beneficial, it's also a matter of being human. We want to know how things work in our subconscious, and the urge to understand that neural networks are our creation is incredibly frustrating. So I believe that as humans, it's intuitive and very understandable that we need to understand how we arrive at decisions to better trust those decisions. And this is part of the human nature. With that, I would like to thank you, and I'm looking forward to your questions and discussion.